Hi everyone, thank you for being here up until the end of the week. This is a joint work that I conducted while a postdoc in the lab of Chaim Wolfson here in the School of Computer Science. And it was done in collaboration with experimental collaborators from the Faculty of Medicine, namely the lab of Mayan Gal, and most importantly, his student, PhD student, Lucy Adriana Lifshitz. Okay, so let me start by defining the terms of the title. Okay, so what are peptides, what are binders, what are peptide binders, why do we care? So peptides are short proteins with a length of fewer than 20 amino acids. In general, a binder is any molecule that binds a target with a protein with high affinity and specificity. So the reason why we are interested in binders is that they can do a lot of things for us. Basically, any drug that we take, most of them are binders. Aspirin, it's a small molecule that binds many enzymes in our body and blocks their activity. When we make an antigenic test, against the corona, it's actually a monoclonal antibody that recognizes, that binds to a protein of coronaviruses and hence enables to detect it. When we go through treatment for, let's say, cancer immunotherapy, then it's also a monoclonal antibody that binds a certain protein along the surface of immune cells, and by doing it so prevents tumor from repressing the immune system by blocking a specific protein-protein interaction. And in general, binders are basically at the heart of modern molecular biology. I can even give you one more example, an example of peptide binder, actually. These very new and exciting drugs for treating obesity and diabetes, these so-called GLP-1 analogs, are short peptides that bind to receptors on the membrane of some of our cells, and these receptors transmit the signal of satiety, basically, such that we do not feel hungry anymore. So generally speaking, binders are of paramount importance in molecular biology. I'll just skip this slide. So I'm sorry, it's an old version. Therefore, we are really interested in designing binders. Designing binders, and in this case, I will talk about peptide binders, that bind a certain target and bind it with high affinity and specificity. So how is it done in general? So consider that we have a certain protein-protein interaction that we want to block. This interaction between here, the red protein and the blue protein. Well, one way to do this using a peptide is to first identify a region along on the red protein that carries out most of the interaction with the blue protein. You can see it here in green. You take off the rest, you just keep the green part, and this is a peptide, a short protein fragment basically, that hopefully still binds the blue protein with high affinity and specificity. In general, you have to further optimize it because as you removed the red part, you lost a lot of strength in binding. So there are usually techniques that are based on experiments, selection experiments, where you conduct mutations of the peptide sequence, and then you select the ones that bind the most to the blue protein. And there are also computational methods that are essentially based on physics. You simulate the physical interaction between your green peptide and the blue protein, and you mutate the sequence in such a way as to further improve the free energy of the interaction. So there are techniques like that that are based on physics. However, in general, the peptide design problem comes with a lot of challenges. There are pros and cons, right? But let's say one of the issues is that, in general, these techniques that are based on physical first principles, they just don't work very well. We don't have good physical models that are computationally tractable and are faithful to, let's say, the truth, the true nature of interaction, which is inherently quantum. And so that's one of the main challenges. Conversely, if you try to do experiments, well, you know, it can be time-consuming. It can it can take a lot of time, of effort, of money, and not end up giving you the result that you want. And also, in general, the space of possible sequences is huge. In general, if I have a peptide of length L, there are 20 possible amino acids, so there's a combinatorial space of 20 to the power L sequences that should be explored in order to find a better binder. Finally, one last issue which is very important is it's not completely clear how to select for additional properties that are really important for therapeutic drugs, like, let's say, bioavailability or toxicity. Okay, so here comes the machine learning side. I'm going to present rapidly an approach for designing peptide binders, which is not based on physics, but based on machine learning, based on basically recognizing patterns that are encountered in evolution. So it's very convenient that I come after Tal, because Tal introduced the concept of protein sequence alignments. In general, there have been in the field, in the last couple of years, very impressive results of designing proteins, so coming up with new sequences that carry out a certain function based on these multiple sequence alignments. And the idea is really the following. These multiple sequence alignments, they are essentially samples in the sequence space. So you can think of it as like a very sparse sampling of this huge combinatorial space. And all these multiple sequence alignments, they correspond to different sequences from other organisms. So you don't necessarily know what these proteins do, but you know that they evolved, that they were selected in order to do this. So the end result is that you can fairly, if there is, let's say, some kind of finesse function that characterizes how well does a protein carries out its function, well, you know that all these sequences, because they have been selected, they are above a certain threshold. And what you're looking for is basically new sequences that lie in the same space, in the space of high fitness function. So one way to do it is to actually fit a function that only using these black points from the data that takes large values next to the black points and low values elsewhere. And the natural way to do it is to fit a probability distribution. So in other words, a generative model that I train on these multiple sequence alignments will learn a probability distribution that is somewhat similar to the underlying fitness function of a problem. And the reason is evolutionary selection, that if there is a sequence that does not perform very well, it will be washed away by evolution, therefore you do not see it. So in practice, it means that what you're looking for is to build a model that preserves, let's say, conservation patterns and coevolution patterns of your data. I will not go too much into details. And there is a very impressive list of experiments that have been successfully, protein design experiments that were based on these techniques. Enzymes and so on. I will not go into the details, but it's incredibly impressive because all of these barbaric names that I'm naming on the screen are very different proteins carrying out very different functions. So what we wanted to do is to apply the same idea for peptide binder design. So we came up with a protocol that is based on this generative model. And the idea is the following. Suppose that we have a target that to which we want to design a binder to, a peptide that binds this with high affinity and specificity. The first thing that we do is we identify all the proteins in nature that already bind this protein. These are its native protein partners. We'll identify them, and we will extract the regions that we know bind this target. The next step is some kind of data augmentation technique, basically, because you only have, at this stage, you only have, let's say, a few dozens of examples. So we're going to use a technique called homology search to search for additional sequences that allegedly carry out the same function based on homology search. Once we have them, we'll build a multiple sequence alignment of these binding fragments. This multiple sequence alignment, we will feed it to a sequence generative model, which will learn the corresponding probability distribution, and then we will draw samples from this generative model. And these samples are candidate binders that allegedly bind or target. So at this stage, you have essentially as many examples as you want. So you have to refine them, to refine your selection. We use both physical modeling and experimental assay to do that. And in the end, you can test your binders in a low affinity setting. So we focused on a target which is called calcineurin. To keep things short, it's a protein that is involved in the response of the immune system. If you are able to block the activity of this enzyme, you can block the cellular immune response. So basically, drugs that target this protein are, for instance, prescribed for preventing rejection of grafts, of transplantations. So so that's the idea. We build a multiple sequence alignments, and now which class of generative model should we use? So to keep, basically, the choice in the end is a bit surprising because we all use a tool which is relatively old, but I just want to give you a reason why. So the model we're looking for has to be suitable for discrete categorical data. So it cannot be something standard like a multivariate Gaussian distribution, and things like GANs or normalizing flows are not suitable as well. It has to take into account correlations between your variables. So a single-site model or an HMM model will not work. It has to be position specific. So because you have these patterns of conservation, for instance, that are really specific on the position. So a model that is translation invariant, like an autoregressive CNN or a recurrent neural network, will actually not work very well. It has to be suitable for very small amounts of data. So anything based on transformers and more complex architecture will not really work as well. And in the end, also, of course, it had to be well established in our field before we started. So new things like fine-tuned generative protein language models and so on didn't exist back then. The answer is what we took is a Markov random field. A Markov random field is a model where fairly old model, things like the POTS model or the restricted Boltzmann machines, which have a very nice likelihood function and can work fairly well even given low amounts of data. So in practice, we use this variant of restricted Boltzmann machine called the compositional restricted Boltzmann machines. It's a graphical model constituted by two sets of coupled random variables, a visible layer and a hidden layer. And basically, the visible layer corresponds to the data, and the hidden layer are latent factors that explains the correlations between your visible variables. So it's very nice because you have a closed form equation for the likelihood function, and you can, which takes this form. You have a linear term over visible layer over your positions on the alignment with GI of VI, and you also have interaction terms that can be written as a sort of a generalized additive model where you have nonlinear functions of projection over the data. So it defines both a representation and distribution over the data, which is really nice. I'm sorry for skipping this slide, but it's an old version. Something happened. Just to keep things short, we validated the model by looking at whether they can predict biophysical properties. And it turns out that if you take your generative model and you calculate you look at the log likelihood function, this function actually correlates with the binding affinity, with the strength of binding of your peptide to your target. So this was really encouraging, and it worked even better than physical models or more elaborate models. So we continued, we generated designs, and we tested them experimentally, and indeed we found that they bound. We found that out of 10 computational designs, 7 of them did bind to the target, including several with high affinity to the data. So that was extremely encouraging. We looked into the details. We also found that the models were quite different from, the peptides we designed were quite different from anything that we found in the nature. Basically, they were obtained by some kind of recombination of natural examples, or they had additional motifs that were not present. And we were extremely happy. So it's very encouraging results for using general generative models in this field of peptide design. There are additional stages that are very important. We don't have a drug yet, of course. You have to solve a lot of additional problems in order to go to something that can be useful in real life. So that's a very important future direction. And we're also looking into algorithmic improvements as well. And one of the directions we're heading to is now to also integrate the structure using generative models. So there are new tools that have been developed that are basically structure-based generative models, where you can essentially find a sequence that falls in a given position. So we're working on combining these evolutionary-based models with structure-based models in order to provide even better designs. So with that, I will conclude that we are very excited with these applications, and we're looking to working on many new targets with new algorithms, and it's a very exciting and ongoing research. Thank you very much.
So I have a question. Do you intend to replace the RBM? Because
sure,
not very fashionable, at least.
Yeah. So the general direction would be to replace it by a prompted generative language model, something that has been pre-trained before, so that it would be suitable to fine-tune on a very small data set, because we don't have a lot of data. And you expect that it can, use previous knowledge. So it's a very important future direction. In practice, it's, I would say, not a very mature field. There are, let's say, generative models that can be take as a prompt a multiple sequence alignment, but there are many practical issues with this. So we look to do this in the future, definitely.
Super cool. Okay, let's thank you.